Good evening, all. I welcome you all for the Reda Academy, your online education partner. And this is session 3 on the chapter 1 Asexual and Sexual Reproduction in Plants from Botany, class 12. So, we are going to continue this lesson as a session 3. Before getting into this lesson, I just first like to summarize you on the previous topics. So this lesson we classified into we just divided them into two half. One is on the asexual type of reproduction carried by the plants and the sexual reproduction in the plants. So in that session 1 we concerned on the asexual type of reproduction carried by the plants. There we studied about the vegetative propagation in detail. In that, natural, conventional and modern methods of vegetative propagation were discussed. And in the second session, we studied about the different events that takes place in the sexual reproduction. We studied about the pre-fertilization changes that is happening in a flower. We studied on the structure of flower, especially we concerned on the androecium, which is called as a male reproductive part, the structure of anther and the structure of pollen grain or microspore, and how they are forming through the process called as a microsporogenesis. And at last we studied about the microgametogenesis, that is the formation of 2-cell and 3-celled stage in the pollen grain. So we stopped up to this in our session 2. So now we are going to see about the megasporogenesis. That is, we are going to study in detail about the female reproductive structure. That is, either we are calling them as pistil or carpel. So this pistil or carpel is the unit of gynoecium. So gynoecium is called as the female reproductive part, and this female reproductive organ is just looks like in a shape of a bottle. It has 3 parts in them. So they are all ovary, stigma, style and ovary. In that, stigma is present at the tip of the female reproductive part, and this stigma is going to receive the pollen grain which is being liberated by the anther during the maturation process. So the anther will stick with the stigma, and later, as we discuss, they are going to produce the pollen tube which contain the vegetative cell and the generative cell. So this stigma is going to receive the pollen grain, and this stigma is connected to the ovary by a long stem like or stalk like structure. We are calling them as a style, and at the end of the style a bulged structure, a Sullivan structure is seen, and that is called as the ovary. And inside the ovary there is a soft tissue structure called as the placenta. In the placenta, the ovules are being embedded. So placenta is the inner structure of the ovary. In the placenta, the ovules are going to attach. So this ovule is called as the female gamete. So you can able to see the ovule which is present in the placenta. So ovule is called as a female gamete. Okay. So now we are going to study in detail about what is the structure of ovule. So as we studied the structure of anther, here we are studying the structure of ovule in the female reproductive part. So ovule is, as I stated, this is called as the female gamete. So this ovule has many parts in them. So the first and foremost part is the funicle, a stalk-like structure which is going to connect the placenta and the ovule. So funicle is a part which is going to connect the placenta with the ovule. So ovule attached with the placenta through this stalk or காம்பு மாதிரியான structure. So this is the funicle region which is going to attach or connect the placenta with the ovule. And the part, the exact point where the funicle is connected with the ovule is called as ileum. So ileum is the point where the funicle joined with the ovule. And there is a long structure called as raphe. So this raphe structure is going to form the sides of the ovule, and they are going to determine the distance between the funicle and the ovule. So raphe is going to determine the distance between the funicle and the ovule. Okay, so this structure which is present at the top, a soft spongy cushion like structure which forms the top of the is actually being inverted. This is an inverted ovule. So chalaza is going to take the basal part. So the basal soft cushion like structure is called as the chalazal region, and they are covered by integuments. Integuments means layers. So there are two layers present in the ovules. One is the outer integument, another one is the inner integument. So we are having two integuments. One is here and second one is here. So two integuments are covering the ovules. So when you are seeing inside this integument, there is a mass of tissue or cells. You are calling them as the nucleus. One of the very important structure of the ovule is the nucleus, because this nucleus are first formed inside the ovule when the ovule is in the immature state. So like the same quet we just studied in the structure of anther, they are having a sporogenous mass of cells present inside the epidermis, and these sporogenous cells will arise as an orchigonial cell. Then they will be dividing and forming all the four layers of anther. Then they are producing the microspore. So this is the outline about the formation of the structure of anther and the formation of microspore. Like the same here, nucleus are going to produce the sporogenous cell. So nucleus are very important as this is going to nourish the embryo, as well as they are going to initiate the structure, or they are involved in the maturation of the ovule. So nucleus is very important structure. And this nucleus, at the center of the nucleus, there is a oval shaped structure. You are calling them as embryo sac. So embryo sac is formed after the cells are getting differentiated in the ovule, and this embryo sac is containing the egg cell which is going to participate in the fusion process. So this is the embryo sac. And the integuments, that is the outer integument and the inner integument, are absent at the region, a small opening like structure which is called as micropyle. So micropyle is the region where the integuments are absent. Okay, so this is some of the important parts when you are taking into concern about the structure of the ovule. So I am not going to tell you what is antipodal cell and secondary nucleus and egg cell, because we have to study in detail when you are studying about the embryo sac. So we have to wait until we are clear about the structure of ovule. Okay, so structure of ovule as the first structure called as a funicle, which is going to make up the stalk-like structure, which is going to present the base. Actually, they are connecting the ovule with the placenta. So funicle, that is the stalk. Okay. Then the integument we told you, that is going to cover them. We have one or two may be present, and depends on the structure of ovule. And we said the helium is the point where the funicle is being attached with the ovule. And Rafi, I told you that the funicle is addend to the body of the ovule that forms a rigid structure which is called as a raphe. And nucleus, I told you, that is actually the central mass of parenchymatous tissue. What is the name of the tissue? They may be asking you in a one-word question. So parenchymatous tissues are the nucleus. So these are going to reserve the food material which is required for the nourishment of the embryo sac and during the development of the embryo. And the micropyle, the pore or opening region where the integuments are absent in them. And chalaza, I told you, that is the basal region of the ovule where the nucleus, integument, funicle, everything meets and merge. So that region is called as a chalazal region which forms a base of the ovule. And the embryo sac or the female gametophyte. So these embryo sac is actually a oval shaped sac like structure which is present in the nucleus and that is going to participate in the fusion of gamete. So this is about the structure of ovule. Some special additional points you can add up when you are seeing on the structure of ovule apart from the parts which we discussed. So the one is that in some of the species, the inner layer, that is we studied about two integument, that is outer integument and inner integument. In very few species, they form unitegamic 10 uninucleate type of integument. That is the inner integument, that is the inner integument which surrounds the nucleus. So that integument will become a specialized structure and they are going to perform some nutritive function for the embryo sac. Such a type of inner integuments are called as endothelium or integumentary tapetum. Tapetum, the word you are aware because நம்ம வந்து antherல discuss பண்ணோம். So inner layer which is going to nourish the microspore. Like the same, the inner layer of the ovule, that is the integument, inner integument, sometime in some of the species become specialized. அவங்களுக்கு special functions இருக்கும். So they will be storing the food material and they will provide the nourishment to the embryo sac. And such a type of integuments are called as endothelium or integumentary tapetum. So the example for this type of special integuments are seen in the Asteaceae family. So there are another, we are just broadly classifying the ovules on this basis we are classifying into two types. One is called as 10 uninucleate type, another one is quasi-nucleate type. So if it is a 10 uninucleate type, here the sporogenous cells is hypodermal with a single layer of nuclear tissue around it. That is when the ovule is in undifferentiated state, that is formation ஆகுது இப்பதான் ovule. So immature stateல இருக்கும்போது they contain the sporogenous cell which are all hypodermal. Hypodermal means they will be present above the layer. So they will be present above the layer and you can see only one layer of the nucleus tissue around them. And such a type of ovules are very smaller in size. Whereas if you take a quasi-nucleate type of ovule, here the ovules will be containing subhypodermal, that is below the hypodermal region, the sporogenous cells will be present. And these type of ovules are larger in size. Okay, so this is going to determine the position of the seed in a fruit, whether it is going to be in a distal end or proximal end. So அதை decide பண்றது எதுனா இந்த type of ovules are going to determine the position of the seed in a fruit. Okay, so there are 6 important type of ovules which we have to know Okay, so let me discuss one by one. First, I like to discuss on the atropus, or you are calling them as orthotropous. That is the first type of ovule. Second type of ovule is the anatropous. Third one is amphitropous. Fourth one is hemiamphitropous, anatropous. And fifth one is campylotropous. And sixth one is crichinotropous. So கொஞ்சம் இது டிஃபிகல்ட்டாக இருக்கும் ப்ரொனவுன்ஸ் பண்ணுறதுக்கு. பட் ஒன்ஸ் யூ நோ த meaning of this type of ovule, then it will be very easy for you people. Okay. So first I told you that is of atropous type. So picture first clear up பார்த்துக்கோங்க. ஒரு definition சொல்லும்போது you just keep the picture in the mind. And here you could see that a micropyle end is present at the top. So you can see the micropyle end at the top and chalazal end is at the bottom. Okay, and you could see that all embryo sac, micropyle end, chalazal all stands in one straight line. Okay, and you could see that the ovule are inverted. தலைகீழ இருக்கு இங்க ovule. Okay, நமக்கு பார்க்கிறதுக்கு இப்படி இருக்கு. Okay, so that is called as atropous, or you can also call them as orthotropous. Okay, so both are like a synonym. You can call it as ortho or atropous. So the micropyle end will be seen at the top and the chalazal end will be at the bottom, and all the three funicle, micropyle, embryo sac, chalazal, all these structure will be stand on a straight line. So that is called as a ortho or atropous. Okay, so let me see here. Orthotropous. Can you able to see? Yeah. So this is a type of ovule where the micropyle is at the distal end. Distal means top. Okay. So the micropyle will be at the distal end, and the micropyle, funicle, chalaza all lies in one straight vertical line. So example for this type of ovule will be seen in the family of Pipereaceae or Polygonaceae. Okay, Pipereaceae or Polygonaceae. And the second type is called as anatropous. Anatropous type of ovules are completely inverted. So usually when you are seeing the picture, இதுதான் தலைக்கீழ இருக்குதுன்னு நமக்கு தோணும். ஓகேவா? பட் எக்ஸாக்டா சொல்ல போனா this is the inverted ovule. எக்ஸாக்டா இந்த இப்படிதான் இருக்கும் ovule பார்க்கறதுக்கு. பட் இதுதான் வந்து inverted structure. பட் நம்ம பிக்சர்ஸ்ல புக்ஸ்ல அடிக்கடி இந்த ஸ்ட்ரக்சர்ஸ் anatropous தான் பார்க்கறதால நம்ம நினைப்போம் இதான் ரைட் ஸ்ட்ரக்சர், இது வந்து inverted. பட் ஆக்சுவலி anatropous is the inverted ovule, not the orthotropous or atropous. So anatropous is the inverted type of ovule. So that you can see the micropylar opening is present at the bottom. So that is present at the bottom, and the chalazal region is present at the distal end. Okay. Yes. Okay. So anatropous, here the body of the ovule become completely inverted. So the micropyle and the funicle lies very close to each other. So you can see that in the picture. So here the funicle is present here. Here the funicle is present here, and you can see that the chalaza, everything are close together. Micropyle, everything are close together. So that's why they are called as a anatropous type of ovule. Mostly if you are taking angiosperms or seeds, அது dicot seed இருந்தாலும் சரி இல்ல monocot seed இருந்தாலும் சரி, angiospermsல வந்து mostly they will be having anatropous type of ovule. And third type of ovule is called as a hemianatropous. Hemi நாலே half அப்படின்றது அர்த்தம். So இப்போ பாதி. Okay. So half anatropous. So that the body of the ovule are placed transversely at a right angle to the funicle. So perpendicular at the right angle they will be. So the example for this type of hemianatropous is the Primulaceae. Okay, we will see. So this is hemitropous. Here you could see that funicle is present here, and you could see the perpendicular right angle present here. Okay, so they are just transversely present. So this is transversely, horizontal present here. Okay, so that is called as a hemianatropous. சுத்தமா complete invert ஆகல, பாதி invert ஆயிருக்கு. So that's why they are called as a hemianatropous அப்படி நம்ம சொல்றோம். ஓகேவா? And the fourth variety is called as campylotropous. So campylotropous of ovule, here the body of the ovule where at the micropylar end is just curved. You can see the curved structure exactly looking like a beans. Okay, or bean or a seed and enna mari curves kidney shape la curved more like the same. You could see the curve or the micropylar end. And the embryo sac is also slightly curved. And the three, that is ileum, micropyle, chalaza, or at the adjacent to one another. ஒன்றுக்கு பக்கத்துல ஒன்று arrange ஆயிருக்கும். And the micropyles oriented towards the placenta. So micropyle will be seen towards the placenta கிட்ட இருக்கும். So that is the campylotropous. Example is a leguminous, that is BOTANY variety. அதெல்லாம் வந்து campylotropusல வரும். Here you could see very clearly on the campylotropous. So this is the funicle region, and you could see the slight curve அப்படி பென்ஸ் தெரியுது பாருங்க. எக்ஸாக்டா இது இங்க இருக்கிற மாதிரி அப்படி ஓவலா அந்த மாதிரி இல்லாம there is a bend is seen on the integument. And also the embryo sac கொஞ்சம் லைட்டா அப்படியே வளைஞ்சிருக்க மாதிரி இருக்கு. ஸோ பிளசண்டா வந்து இங்கே தானே இருக்கும் ஃபினிக்கல் அது அட்டாச் ஆகிற இடத்துல. ஸோ ஃபினிக்கலுக்கு ரொம்ப கிட்டத்துல தான் இந்த மைக்ரோபைல் ஹிலியம் பார்த்தா ஆல்மோஸ்ட் கர்வ்ட் அண்ட் பெண்ட். ஸோ டுவர்ட்ஸ் அந்த பிளசண்டாக்கு ஓரியன்டாகவே எல்லாமே கிட்டவே இருக்கும். ஸோ தட் இஸ் கால்ட் அஸ் அ campylotropous type of ovule. Okay, and next one is the amphiotropous. So amphi அப்படினாலே you know that it is having both the character. So the distance between the helium and the chalaza is very less. So the curvature of the ovule will be clearly exhibited, and it looks like a horseshoe shape. இந்த மாதிரி இருக்கும். This is horseshoe shape. So the amphiotropous will have a very lesser distance between the helium and the point, helium point and the chalaza, and you could see the horseshoe shaped structure. And this type of amphiotropous ovules are seen in the Alismataceae. Okay, so here is the amphiotropous. So in the amphiotropous you can clearly see that the helium பாருங்க எப்படி இருக்கு. Okay, so helium is just too closer, and you can see the curvature structure. Okay, so this is how they are. So that is why they are called as a amphiotropous. And the last type is called as a cricanotropous. So in the cricanotropous, funicle is very long and surrounds the ovule. Funicle எவ்வளோ லாங்காக இருக்கும்னா ovule சுற்றி வர அளவுக்கு ரொம்ப லாங்காக இருக்கும். So that type of ovule is called as a cricanotropous. Example is cactaceae. So here you could see that. Funicle பாருங்க. Just a minute. Yeah. So funicle you can able to see. இங்க இருந்து ஆரம்பிச்சு அப்படியே ஃபுல்லா கவர் பண்ணிடும். So they are covering the even the ovule. Ovule அப்படியே ஃபுல்லா கவர் பண்ற அளவுக்கு funicles are very longer. So these are the 6 type of ovule. So to keep in memory very easily, ortho or atropous type of ovule is just micropyle at the top, and anatotropous micropyle is at the bottom, and amphiotropous has a curvature structure. Anatropous, they will be transversely perpendicular. Campylotropous has lots of bend bean shaped structure will be seen in the campylotropous, and cricanotropous where the funicle is very long and covered the ovule. So இப்படி தான் ஞாபகம் வச்சுக்கணும். ஓகேவா? So these are the different types of ovule. So after we are seeing the structure of the ovule, now we are getting into how the female gametophytes are going to produce. As I told you earlier, micro means male, mega means female. So megasporogenesis refers to the formation of megaspore. That is the megaspore mother cell, MMC. அங்கே அந்த மாதிரி MMC படிச்சிருப்பீங்க. microspore mother cell. So that formation of this megaspore mother cell is called as a megasporogenesis. And we are going to see how that is going to form inside a ovule. So as the ovule develop from the immature state or mature state develop ஆகும்போது a single hypodermal cells in the nucleus become enlarged. I told you already nucleus are very very important because that is going to differentiate and they are one who is going to form the megaspore mother cell. So inside the ovule structure integuments இருக்கும் integumentsக்கு ஃபுல்லா உள்ள என்ன இருக்க போகுது nucleus தான் because it is a immature um, ovule. So there is no differentiation of embryo sac அதெல்லாம் எதுவுமே இருக்காது. Just a mass of cells will be present in them. At the single hypodermal cells will be present there. So this is called as a nucleus. You are calling them as a nucleus. In that nucleus, then there will be enlargement. ஒரு சில cells மட்டும் நல்ல பெருசா enlarge ஆயிடும். So that cells you are calling it as the archisporium. So that type of cell is called as a archisporium. So this archisporial cells may directly function as a megaspore mothers. எந்த விதமான divisions எதுவுமே இல்லாம directly they will be acting as a megaspore mother cell and start producing the megaspore sometimes. In other situation, they goes for a transfer division. அதாவது இப்படி ஒரு ஆர்கிஸ்போரியம் செல் இருக்குன்னா they will be dividing like this. Transversely they are dividing and they will become two archisporial cells. Okay, so these two archisporial cells are called as the primary sporogenous cell or you are calling it as primary parietal cells. ஒன்று வந்து primary parietal cell, this is forming the outer layer and another cell is forming the inner primary sporogenous cell. Either the archegonial cell will directly act as a megaspore mother cell or they will go for the transfer division. In that transfer division, one which will be forming the outer layer, that is primary parietal cells, the outer layer, and the one will be forming the inner mass of primary sporogenous cell. இப்போ நம்ம வந்து மைக்ரோஸ்போரோஜெனசிஸ் படிக்கும் போது இதே கான்செப்ட் தான் நம்ம படித்தோம். ஸோ உள்ள இருக்கிற ஸ்போரோஜெனஸ் செல்ஸ் ஆர் ப்ரீக்ளினலி அண்ட் ஆன்டிக்ளினலி டிவைடிங். சம் ஆஃப் தேம் ஆர் கோயிங் டுவர்ட்ஸ் த எபிடர்மிஸ் and forming all the three layers. What are they? Endothelium, middle layer and tapetum form. So inner cleavage ஆகி போகிறது எல்லாமே மைக்ரோஸ்போர் ஃபார்ம் பண்ணணும்னு பார்த்தோம். அதே மாதிரி தான் இங்கேயும் மெகாஸ்போரோஜெனசிஸ்ல. So they are going to divide under two portion. Outer portion divide ஆகிறது எல்லாமே they are going to produce the primary parietal cell and innerly they are dividing to form the primary sporogenous cells. So this parietal cell, primary parietal cell, they remain undivided or sometimes they divide either by preclinal or anticlinal division and they embed the primary sporogenous cell deep into the nucleus. So தொடர்ந்து இந்த parietal cells divide ஆயிட்டே இருக்கிறதால sporogenous cells இவங்க என்ன பண்றாங்க, புஷ் பண்றாங்க. எதை நோக்கி? நியூசிலஸ்குள்ள புதைக்கிற அளவுக்கு புஷ் பண்றாங்க. தென் இந்த மாதிரி புதைஞ்ச அந்த sporogenous cells தான் then they are later developing into the megaspore mother cell. Okay, so the primary sporogenous cell I told you, so which is being functional as a megaspore mother cell. So இந்த மாதிரி நியூசிலஸ் நோக்கி புஷ் பண்ணப்பட்ட அந்த primary sporogenous cells தான் என்னவா மாற போறாங்க, megaspore mother cells ஆ மாற போறாங்க. Then this megaspore mother cell are undergoing meiotic cell division because the gamete form பண்ண போதுனாலே reduction division நடக்கும், meiotic cell division, and they are going to form 4 haploid megaspore. அங்கேயும் நம்ம அதுதான் பார்த்தோம். அங்கேயும் வந்து மைக்ரோஸ்போர் மதர் செல் வந்து they are going to divide into tetrad. 4 pollen grains divide ஆகும், மைக்ரோஸ்போர்ஸா divide ஆகும் நம்ம பார்த்தோம். Like the same, the megaspore mother cell will undergo for the meiotic division to form a 4 haploid megaspore. In that 4 haploid megaspore, only one is going to develop and other 3 will get degenerated. Okay, so மொத்தம் 3 megaspore, I mean 4 tetrad இல்லையா? ஸோ இந்த மாதிரி 4 வந்து ஃபார்ம் ஆகும். யூஸ்வலி இவங்க வந்து லீனியர் டெட்ராய்ட் தான் ஃபார்ம் பண்ணுவாங்க. மைக்ரோஸ்போரோஜெனிசிஸ்ல அப்படி இருக்காது. இந்த மாதிரி கிளஸ்டர்டு டெட்ராய்ட்ஸ் இப்படி இருக்கும் டெட்ராய்டு. இதில் வந்து லீனியராக இருக்கும் டெட்ராய்ட், ஒன்றுக்கு அப்புறமா ஒன்று இந்த மாதிரி. இதில் வந்து செலேசல் எண்டில் இருக்கிற 3 megaspores may degenerate and they will not be going to form the megaspore. So only one will survive and that survival is called as a monosporic development ன்னு சொல்றோம். So ஒன்னே ஒன்று மட்டும்தான் functional megaspore மாறும், that is female gametophyte develop ஆகும் embryo sac குள்ள. அப்போ ரிமைனிங் இருக்கிற 3 என்ன ஆகும்னா they will get degenerated, அப்படியே dissolve ஆயிடும். So that is why we are calling them as a mono. Mono means single. So only one spore is developed out of the 4 divided cells. So that's why we are calling them as a monosporic development. Example, polygonum. Sometime it may be bisporic, sometime it may be tetrasporic. சில மெகாஸ்போரோஜெனிசிஸ்ல 4 வந்து 2 மட்டும் ஃபங்க்ஷனலாக மாறும். சில டைம்ல 4மே ஃபங்க்ஷனலாக மாறும். இட்ஸ் டிபெண்ட் ஆன் த டைப் ஆஃப் ஓவியூல், ஓகே, அண்ட் டைப் ஆஃப் பிளான்ட். ஸோ usually 70 to 80% of the megasporogenesis leads to the formation of monosporic development. That is only one will be functional, other will not get disintegrated. So இப்போ நான் சொன்னது அப்படியே பிக்சர்ஸ்ல காமிச்சிருக்கேன். You can able to see that. Here this is called as a megaspore mother cell. First meiotic division அப்ப. So இப்போ for example இதில் வந்து 12 number of chromosome இருக்குன்னா 6 அப்படின்னு டிவைட் ஆக போகுது. ஓகே, ஃபர்ஸ்ட் meiotic cell division. Then second meiotic cell divisionல they are just going to form. இங்க வந்து all this going to have a 6. That is second meiotic cell division. This is called as a equational division. ஏன்னா இங்க என்ன நடக்கல, ரிடக்ஷன் நடக்கல, அப்படியே எக்ஸாக்ட் காப்பி வந்து ஃபார்ம் ஆகுது. ஆனால் இது மியாசிஸ் ப்ராசஸ். ஸோ யூ ஆர் காலிங் them as a equational meiotic division. So 2 become 4 marriage. Okay, so all are having haploid number of chromosome n. They are all having a haploid number of chromosome. Out of this 4 megaspore, the 3 will get degenerate. 3 என்ன ஆயிடுச்சு, degenerate ஆயிடுச்சு. So only one get enlarged and this is called as the functional megaspore. So you are calling it as a functional megaspore அப்படின்னு நம்ம சொல்றோம். ஸோ ஒன்று தான் சர்வைவ் ஆகுன்றதால இதுக்கு பேர் monosporic development அப்படின்னு நம்ம சொல்றோம். So now they are forming the one megaspore out of 4. So now the megaspore is ready. Okay. So now this megaspore is going to, as I told you, see here, I had given some example for the bisporic. Allium is an example for bisporic and tetrasporic example is the peperomium. So here all the 4 megaspores will be developed as a functional megaspores. So then the ovule generally has a single embryo sac. எல்லா ஓவியூல்ஸ்மே ஜென்ரலாக ஏஞ்சஸ்போம்ஸோட எல்லா ஓவியூல்ஸுமே எடுத்து பார்த்தீங்கன்னா உள்ள ஒரே ஒரு எம்ப்ரியோ சாக் தான் இருக்கும். ஓகே, அந்த எம்ப்ரியோ சாக்குள்ள ஒரே ஒரு மெகாஸ்போர் தான் இருக்கு, பிகாஸ் மிச்சம் 3 டீஜெனரேட் ஆயிடுச்சு. ஸோ they are having a functional, only one functional megaspore which will be formed as a embryo sac or a female gametophyte. So this megaspore is going to elongate and they are going to divide by the mitotic division. So one அப்பா as the இப்போ ஒரே ஒரு மெகாஸ்போர் தான் இருக்கு, அது வந்து என்ன பண்ண போகுது, elongate, இது மாதிரி elongate ஆக போகுது. Then they are going to divide and one will be going to the chalazal region, one will be going to the micropylar region. So ஒரு செல்லா இருந்தது இப்போ என்னவா மாறிடுச்சு, மைட்டாட்டிக் செல் டிவிஷன்ல 2 செல்லா மாறிடுச்சு. ஓகேவா? So the one will be found in the micropylar axis and the other will be found on the chalazal axis. And after this nucleus are going to the both the pole, the walls doesn't form. So அவங்க வந்து அவங்க நியூக்ளியஸா மட்டும்தான் இருப்பாங்களே தவிர செல்லா மாற மாட்டாங்க. எப்பவுமே ஒரு செல் டிவைட் ஆகுதுன்னா 2 ஸ்டேஜஸ் இருக்கு. ஒன் இஸ் கால்ட் as the karyokinesis Another one is called as the cytokinesis. So karyokinesis na முதல்ல nucleus வந்து என்ன ஆகணும் divide ஆகணும். So that is called as karyokinesis. That is, so இப்போ செல்லுக்குள்ள ஒரு nucleus first என்ன ஆகும்னா nucleus தான் ரெண்டா divide ஆகும். So that is called as karyokinesis. After the karyokinesis, the cytoplasm will start divide and they will be forming two cells. அப்ப செல் வால் ஃபார்ம் ஆகணும் இல்லையா? சைட்டோபிளாசம் சுத்தி செல் வால். இப்பதான் இவங்களை நீங்க என்னன்னு கூப்பிடுவீங்கன்னா செல்லுன்னு கூப்பிடுவீங்க. அப்போ இந்த ஸ்டேஜ்ல பாத்தீங்கன்னா only nucleus are divided. So you cannot call them as a cell. You are calling them as only nuclei. So இந்த term ரொம்ப ரொம்ப இம்பார்ட்டன்ட். ஏன்னா நம்ம எம்ப்ரியோட அப்பரேட்டஸோட ஸ்ட்ரக்சர் படிக்கும்போது நியூக்ளியைங்கிறது வேற டேர்மு செல்லுங்கிறது வேற டேர்ம். ஸோ கன்ஃபியூஸ் பண்ணிக்க கூடாது. ஸோ நியூக்ளியஸ் டிவைட் ஆனாலும் அவங்க என்னவா இருக்கிறாங்கன்னா நியூக்ளியா தான் இருக்கிறாங்க பிகாஸ் தே ஆர் நாட் சரௌண்டட் பை த செல் மெம்பரேன். ஒன்ஸ் இட் இஸ் சரௌண்டட் பை த செல் மெம்பரேன் தென் தே வில் பிகம் அ கம்ப்ளீட் செல். Okay, so as walls are not formed, they are just a nuclei. One nuclei went to the micropylar end and the nuclei went to the chelazal end. So two daughter nuclei are formed. Okay, and now as the vacuoles which is present the centrally they are having a vacuole. Central large vacuole will appear between these two daughter nuclei. They are forming a large vacuole. So this large vacuole is going to push the nuclei to the two polar ends. Okay, so they are going to push the daughter nuclei to the two polar that is two poles, opposite poles of the embryo sac. So ரெண்டு போலுக்கு வந்து அவங்க போயிட போறாங்க. Okay, so one to the chelazal pole, another one is to the micropyle pole. So after they are reaching the pole, again they are going for the second mitotic division and then third mitotic division. So second mitotic division வரும்போது they will be forming two cells. So one cell வந்து இங்க 2 மாறும், அதே மாதிரி இங்க வந்து one cell வந்து 2 மாறும். இது வந்து second mitotic division. அதுக்கப்புறம் மறுபடியும் ஒரு third mitotic division போகும்போது they will be forming 4 cell at the chelazal region and 4 cell at the micropyle region. மொத்தம் 8 நியூக்ளியை வந்து என்ன ஆயிடுச்சு இப்போ ஃபார்ம் ஆகி ரெடியா இருக்கு. ஓகே, so as twice mitotically they are forming 4 nuclei at each pole. Totally we are having 8 nuclei now. Okay, so ஒரே ஒரு மெகாஸ்போரா இருந்தது என்ன ஆயிருக்கு டிவிஷன் ஆகி டிவைட் ஆகி now we are having 8 nuclei, but that all 8 nuclei is present only inside the embryo sac. ஒரே ஒரு embryo sac தான், அதுக்குள்ள இப்போ 8 nuclei வந்து இருக்கு. Now these nuclei are going to arrange in a pattern. ஒரு பேட்டர்ன்ல அரேஞ்ச் ஆக போகுது and சில nuclei வந்து செல்லா மாற போறாங்க by forming the cell membrane. Others that just going to remain as a nuclei. Let me see how they are going to arrange. இது பிக்சர்ல காமிச்சா உங்களுக்கு கரெக்டா புரியும். Okay, here. So I told you only one megaspore is become functional. Other this is called as a megaspore mother cell. First meiotic cell division and second meiotic cell division. ரெண்டு செல் டிவிஷன் நடந்து முடியும் போது 4 megaspores are formed, tetrads. So they are arranged in a linear fashion. Okay, out of the 4, 3 get degenerated, only one is remained, and this megaspore or embryo sac is haploid in nature. And they go for the first mitosis and they are forming 2 nuclei. After the 2 nuclei are formed, because of the vacuole, they are pushed to the poles. So this is the chelazal pole and this is the micropyle pole. So இந்த ரெண்டு போலுக்கு அது push ஆயிடும். Then they will be going for the second, third mitotic division and they are forming 8 nuclei. How many nuclei? 8 nuclei are formed at the end of third mitosis. Then after the formation of 8 nuclei, 3 of them are present at the chelazal end, 3 of them are present at the micropylar end, 2 of them are present at the center region. அந்த மாதிரி அரேஞ்ச் ஆயிரும் 3 2 3 அப்படி ஃபார்ம் பண்ணிடுறாங்க. So when they are being arranged in such a way, so the 3 which is present in the chelazal end, you are calling them as antipodal cells. ஏன் இப்போ இவங்களை செல்லுன்னு கூப்பிட்டுட்டோம்? Because these nuclei are surrounded by the cell membrane. Cell membrane வந்து கவர் பண்ணி இவங்களை ஒரு செல்லா மாத்திடுச்சு. ஸோ நியூக்ளியா இருந்தது என்னவா மாறிடுச்சு, இப்போ செல்லா மாறிடுச்சு. ஓகேவா? ஸோ they are surrounded by the cell membrane and become a cell. So there are 3 cells present at the chelazal end, and you are calling these 3 cells as antipodal cells அப்படி நம்ம கூப்பிடுறோம். ஓகேவா? and then இங்க மைக்ரோபைலர் எண்ட்ல பார்த்தீங்கன்னா இங்கே எத்தனை செல்ஸ் இருக்கு? 3 செல்ஸ் இருக்கு. இங்கே நம்ம அவங்களை என்னன்னு கூப்பிடுறோம்னா செல்லுன்னு கூப்பிடுறோம். பிகாஸ் தே ஆர் சரௌண்டட் பை த மெம்பரேன். ஸோ இங்கே ஒரு நியூக்ளியர் இருக்கு பாருங்க. 1 2 3 3 நியூக்ளியர். இந்த 3 நியூக்ளியுமே என்னென்ன சரௌண்ட் ஆயிடுச்சு? நியூக்ளியர் மெம்பரேன், ஐ மீன் செல் மெம்பரேன் சரௌண்ட்ஸ் த நியூக்ளியர். ஸோ தீஸ் 2 ஆர் கால்ட் அஸ் சினர்ஜிட்ஸ். சினர்ஜிட் செல்லுன்னு சொல்லுவோம். ஏன்னா அவங்களோட ஸ்ட்ரக்சர் பாருங்க, ஃப்ளாஸ்க் மாதிரி இருக்கு பார்க்கறதுக்கு. ஓகே, ஸோ they are in a structure of a flask and they are going to take the micropylar end and they are producing a filiform apparatus. Very 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 important, because this filiform apparatus is going to produce a chemical substances which is going to give a signal to the pollen tube. Pollen tube are blind, right? They have to get into the style and they have to reach the embryo sac. For that they require some chemical signal. So that chemical signal will be given by the filiform apparatus. அப்போ அந்த pollen tubeல இருக்கிற vegetative cell இந்த சிக்னலை கரெக்டாக கேட்ச் பண்ணிட்டு கரெக்டாக அழகாக என்ன பண்ணுவாங்க, ஸ்டைல் வழியா உள்ள டிராவல் பண்ணி எக்ஸாக்டாக மைக்ரோபைலர் ரீஜனை ரீச் பண்ணுவாங்க. ஸோ இவங்க 2 பேருக்கு நடுவில் இருக்கிற interaction தான் creating the pollen pistil interaction. So synergids are very important, and the one which is present at the top of the synergid is called as a egg. You are calling it as a egg. So this egg is haploid in nature and they are the one who is going to fuse with the male gamete. இவங்க தான் மேல் கேமெட்டோட ஃபியூஸ் பண்ணி ஜைகோட்டை ப்ரொடியூஸ் பண்ண போறவங்க யாருன்னா இந்த எக் தான். ஓகே ஃபைன். ஸோ இப்போ இந்த எண்டில் வந்து மொத்தம் 4 இருக்குது. இங்கே பார்த்தீங்கன்னா தெரியும் 4 நியூக்ளியை இருந்துச்சு. அதில் 3 நியூக்ளியை வந்து செல் மெம்பரேன் ஃபார்ம் பண்ணி செல்லாக மாறி இந்த செலசல் ரீஜனுக்கு போயிடுச்சு. அப்போ இந்த ஒரு போலர் நியூக்ளியக்கு என்ன ஆச்சு? இது என்ன ஆச்சுன்னா தே ஆர் மூவிங் டுவர்ட்ஸ் த சென்டர். இங்கே சென்டரை நோக்கி மூவ் பண்ணி வந்துருச்சு. அண்ட் தென் இங்கே பார்த்தீங்கன்னா அதே மாதிரி 4 நியூக்ளியை இருக்குது. out of which 3 are being surrounded by the cell membrane and they will become 2 synergid 1 egg cell மாறிடுச்சு. அப்போ இந்த ஒரு நியூக்ளியக்கு என்னாச்சு? இதுவும் வந்து சென்டரை நோக்கி travel பண்ணிடுச்சு. and centrally they will be present and they are all called as the polar nuclei அப்படி நம்ம சொல்றோம். because they are still in a nuclei condition, because they are not surrounded by the cell membrane separately. Okay, so now they are all antipodal region இருக்கிற 3 செல்லுமே they are all N applied இருக்காங்க. அதே மாதிரி synergidsம் N applied இருக்காங்க. எக்கோ N. பட் போலார் நியூக்ளியில 2 நியூக்ளியே இருக்கு. அதனால போலார் நியூக்ளியை என்ன கண்டிஷன்ல இருக்காங்க? diploid condition. So the polar nuclei is in the diploid condition. So in the stage now the female gamete is ready and you are calling this as the egg apparatus. என்னன்னு கூப்பிடுறோம் அவங்கள நம்ம egg apparatus. பார்க்கறதுக்கு வந்து ஒரு முட்டை மாதிரியே இருக்கு இல்லையா? அந்த முட்டைக்குள்ள இந்த செல்ஸ் எல்லாம் என்ன ஆயிடுச்சு? கரெக்டாக எக்ஸாக்டாக ஒரு ஃபார்மேட்டுக்கு வந்து நின்றுட்டாங்க. ஓகே, ஸோ megasporogenesis got over. Now the female gamete, that is embryo sac, அதுக்குள்ள egg cell polar nuclei ரெடியாக இருக்காங்க ஃபியூஷன் ப்ராசஸ்க்காக. already microsporogenesis முடிஞ்சிச்சு. Then the pollen grain are now being deposited on the stigma by the process called as pollination, and they are going to produce the pollen tube. So everything is ready. So pre-fertilization event get over. So gametogenesis முடிஞ்சிச்சு. Male gamete pollen grain has been formed, and female gamete the ovule has been formed and ready. So now the second step is the gamete transfer. So the male pollen grain has to transfer from the anther to the stigma. So in the process we are calling it as a pollination process. So in the pollination process and how it is happening, what are the different agents that is responsible for the pollination, and what is the result of pollination, pollen pistil interaction. These are all we will study in the upcoming session. Session 4 la pollination patti detailly let we see. Okay, and after that we will getting into the second event of the sexual reproduction, fertilization or post fertilization changes yo session 5 la vandu நம்ம பார்க்கலாம். Okay, so I think now you get a very clear picture about the megasporogenesis. So microsporogenesis we studied in session 2. Session 3 concern only on the megasporogenesis, and session 4 the upcoming session will concern on the pollination, types of pollination, how the pollination is going to happen, what are the agents that are controlling the pollination, that we are going to see in the session 4. And it will take one more session, that is session 5, for the second event, fertilization and post fertilization changes. Maybe 2 more sessions also needed, I think so. 5, 6, 7 even it takes, because this is such a very long process and you should have a detailed view on this, because weightage in the lesson அதிகம். So நிறைய questions கேட்பாங்க, twisted கேட்கறதுக்கு நிறைய chances இருக்கு. So you have to get ready with this lesson. So I am just going it section by section, ஒரு டாபிக்கா எடுக்கிறேன் நான். ஏன்னா ஒரே நேரத்தில் எல்லாத்தையுமே புஷ்அப் பண்ணால் you can't able to get it up. So we are just going step by step. So it will be very easy for you people to prepare for the exams. Okay. So thank you all. Let me meet in the next sessions discussing on the pollination. Thank you.